Welcome to The Explainer. I'm your host, and today we are taking a seriously fascinating journey inside your own body. We're going to explore the cellular economy. Think of it as the hidden microscopic hero's journey of a single glucose molecule as your body transforms the food you eat into vital cellular energy.
So, picture a puffin diving into the freezing ocean to catch a sand eel. How on earth does that puffin power its flight, keep its heart beating, and, well, sustain its life just by eating those tiny fish? More broadly, how does organic fuel actually translate into cellular work? It's a fundamental problem of biology, and the answer is incredibly elegant.
Well, it all comes down to cellular respiration. By definition, this is the aerobic process where cells harvest chemical energy from organic molecules to generate a little something called ATP. You can think of ATP as the critical biological drive shaft that links catabolism to cellular work. It makes absolutely everything happen in the cell. It is literally your universal energy currency.
Okay, let's lay out the roadmap for today. We'll start with the cellular energy problem, figure out how to split the sugar in glycolysis, ride through the citric acid cycle, cash in our energy during oxidative phosphorylation, and finally, we'll tackle the oxygen problem.
Number 1, the cellular energy problem. You see, the food you eat contains massive amounts of potential energy locked in its chemical bonds. But releasing it all at once, that would be a complete disaster. If a cell released all the energy from a fuel, like glucose, in a single step, it would cause a damaging release of heat and light. Basically, a microscopic explosion right there in the cell. It'd be like a gas tank exploding instead of powering a car engine in controlled, steady steps.
To extract this energy safely, the cell relies on redox reactions. There's a key contrast here. Oxidation is the loss of electrons from a substance, while reduction is the addition of electrons to a substance. And these two, they always go hand in hand. By moving electrons from one molecule to another, specifically towards highly electronegative oxygen, the cell safely transfers energy downhill in small, totally manageable steps.
To really wrap our heads around this, let's look at a brilliant analogy from our source material. Imagine you're holding a crisp $100 bill. That's your glucose molecule. It holds a ton of value, right? But you can't easily use it to, say, buy a pack of gum from a vending machine. Respiration is exactly like cashing that $100 bill at an ATM for a stack of highly usable $1 bills, which represent our ATP. It solves that explosive energy problem by systematically cashing in large fuel molecules for smaller, highly usable energy units.
So how exactly do we get those $1 bills? It happens in 3 main stages. Step 1 is glycolysis. Step 2 involves pyruvate oxidation and the citric acid cycle. And step 3 is oxidative phosphorylation. Lock this mental map in your mind, because we're going to refer back to it constantly as we track our glucose molecule's journey through the cell.
Number 2, glycolysis, splitting the sugar. We are now in the cytosol, the fluid of the cell, where our glucose molecule faces its very first major transformation. Now, glycolysis literally translates to sugar splitting. By definition, it's the initial breakdown of a 6-carbon glucose molecule into 2 3-carbon molecules called pyruvate. And what's absolutely incredible about this process is its evolutionary age. Because it happens right in the cytosol and doesn't need any fancy membrane-enclosed organelles. It's an ancient metabolic heirloom from early cells. And it occurs whether oxygen is present or not.
Glycolysis operates in 2 distinct phases. First, the cell actually has to spend 2 ATP in the energy investment phase. You know, it takes money to make money. But it definitely pays off in the energy payoff phase, producing 4 ATP. This gives us a modest net yield of 2 ATP, 2 pyruvate molecules, and importantly, 2 NADH molecules. Think of NADH as little shuttles that have captured high-energy electrons for later use.
Number 3, the citric acid cycle. Now we track those newly split pyruvates as they plunge into the mitochondrion for the next intense stage of energy extraction.
Following the timeline here, before entering the cycle, the pyruvate must be prepped. In step 1, it actively transports into the mitochondrion. In step 2, a multi-enzyme complex strips off a carbon, releasing the very first bits of carbon dioxide and attaches a coenzyme to form acetyl-CoA. Finally, in step 3, this high-energy acetyl-CoA enters the biological furnace known as the citric acid cycle.
So what exactly comes out of that furnace? Well, for every acetyl group that enters, the cycle completely breaks it down, outputting 3 NADH, 1 FADH2,
1 ATP and 2 CO2 molecules. And here's a mind-blowing fact to connect this to your own life. This exact microscopic cycle is what produces the carbon dioxide that you are exhaling with every single breath right now. Pretty cool, right?
Number 4. Oxidative phosphorylation and ATP. We've reached the climax of our journey. But we have a new problem to solve. How do we actually cash in all those high-energy electron shuttles we've been hoarding?
The answer is chemiosmosis, defined as the use of a hydrogen ion gradient across a membrane to drive cellular work. Think of it like this. The electron transport chain breaks the fall of electrons to oxygen into manageable steps. As electrons fall, kind of like gravity, down this chain, their energy is used to pump protons across the mitochondrial membrane. Those protons then rush back down their gradient through a massive rotating enzyme called ATP synthase. It's exactly like a rushing stream of water turning a physical water wheel, mechanically generating ATP from ADP.
So what's the grand total payout? About 32 ATP. This massive windfall comes from just a single glucose molecule, showing the incredible power of oxidative phosphorylation. It takes the relatively small amounts of ATP we generated earlier and multiplies them massively by finally cashing in all those electrons we've been so carefully collecting.
To put this into perspective, let's talk efficiency. Cellular respiration transfers about 34% of the potential chemical energy in glucose directly into ATP. Now, you might hear 34% and think, that sounds kind of low, but biologically speaking, it is remarkably efficient. For comparison, even the most efficient automobile engine only converts about 25% of the energy stored in gasoline into motion. The rest of the energy from glucose is released as heat, which, by the way, is exactly how we maintain our 37 degrees Celsius body temperature.
Number 5. The oxygen problem. Now let's tackle our final hurdle. What happens when a cell's system lacks the crucial oxygen needed at the very end of the electron transport chain? Well, without electronegative oxygen there to pull those electrons down the chain, the whole oxidative phosphorylation factory literally grinds to a halt.
When oxygen is absent, cells pivot to fermentation, taking one of two common paths. Lactic acid fermentation is used by human muscle cells when you're working out hard and the oxygen from your blood is scarce. It's also used by certain fungi and bacteria in the dairy industry to make cheese and yogurt. On the other hand, alcohol fermentation is used by yeast. It reduces pyruvate to ethanol and releases CO2, which is exactly what makes baker's bread rise and brews our beer.
The core takeaway here is that both of these processes operate entirely without oxygen. Their main biological goal actually isn't to make massive amounts of energy. It's to recycle those NAD+ electron shuttles so that the basic glycolysis cycle can keep running. It yields only 2 ATP per glucose molecule, so while it's vastly less efficient than aerobic respiration, it's an absolutely crucial biological backup plan. It's the emergency protocol that keeps cells alive when they are practically suffocating.
We'll end on this fascinating puzzle. If respiration and ATP production are so crucial to life, why do hibernating mammals intentionally reduce their ATP efficiency during the winter? Well, they have specialized brown fat cells packed with mitochondria. But these mitochondria have a unique uncoupling protein that allows protons to flow back across the membrane without turning that ATP synthase water wheel. Instead of making ATP, the energy from burning fat is released purely as heat. They intentionally reduce their metabolic efficiency to generate life-saving warmth during the freezing winter without building up toxic levels of unused ATP. It leaves you marveling at biological adaptability, doesn't it? So it makes you wonder, what other incredible survival mechanisms might be hiding in the fundamental chemistry of our cells?